But in reality, it's like going from, making pepperoni pizza one day to making salted caramel ice cream the next day. Similar types of problems, but really you can't learn anything from the pizzas that applies to the ice cream.
Well, you certainly wouldn't want to take your temperature setting and apply it from one to the other.
Right, yeah.
You'd end up with ice cream soup pretty quickly. Boiling ice cream soup. Oh, that doesn't sound good. Today, understanding complex living systems is hard. But does it have to be? I'm Marcus, I'm a biologist.
I'm Phil, I'm a statistician. Together, we discuss the best experiments to cut through biological complexity. Join us to explore the shape of the next experiment. What's the ultimate way that we might explore biological complexity? So we know that multi-dimensional experiments are powerful for optimizing individual problems, individual projects, but we must be amassing more and more of these data sets, these multi-dimensional data sets. So is there a way that we can fully exploit these, better in aggregate and maybe get even deeper insights into biology? That's what I think we want to talk about in this episode. So I think you've got some fairly starry-eyed ideas about how this might work.
Yeah, so I've got this notion like you say, we know that as we do multi-dimensional experiments, then we can solve a particular problem, right? So we can apply it to doing assay development in early discovery, say, and we can develop fantastic assays really quickly and really understand the factor space and the overall assay possibilities a lot better than we'd otherwise would. But then it feels like, okay, so a typical pharma company might be doing 70 assays a year, right? And so now they've got 70 sets of multi-dimensional data covering all sorts of different assays. So surely there must be a way that we can use those high quality, high dimensional data sets to inform us about the 71st and the 72nd. And I feel like this is where AI comes in.
Right.
Mm-hmm. So, where we've seen AI effectively applied in biology is often where you have these big data sets and you can essentially rely on it to be able to see across the scope of all of those data sets and to pull out areas where there could be correlations or patterns. And essentially, this has been used in various things like identifying new targets, which could be for treating particular diseases or whatever. Like it's been used fairly effectively. And it just gives me this sense that maybe, this sort of utopian vision, if you like, of where not only are we solving each problem as we come, but then we're adding to this aggregate data set and the analysis of all of those data set, that meta-analysis, which means that then every experiment we run is from a higher basis of knowledge, right? Mm-hmm. Like that combination of artificial intelligence, human intelligence, such that every experiment you're doing is starting from a higher place in the first place. And like that seems just like a logical progression to me.
Yeah. If I was being unfair, what I might say is that, we hear AI used often in these situations where it's like, there's this very tricky problem.
Right.
The magic of AI is going to solve it.
Right.
So I think, we need to probably delve into this a bit more and to really understand how is that actually going to work,
Yeah, I think, there's that classic thing, so it's all about the data that you train the model on, right? Mm-hmm. AI, it's a problematic term.
Yeah.
Because it implies some kind of magic, right? Yeah. And there is no magic, right? Sorry to disillusion anybody who might be listening, but it's more that, and I think it's worth kind of just replacing the term AI for that of software. Right. So it is still just software, but it's software that's very capable of teasing out correlations and patterns where they are there. But then it depends very much on the data we're feeding it, right? And maybe, in the same way as when we're thinking about doing an optimal experimental design, we're thinking about, okay, how are we designing this experiment to build a particular model? Then if we are to use data in this way, then we can't just be thinking about the experiment today.
right.
when we're doing an experiment.
Yeah.
Maybe we also need to be thinking about that it might be used in future, for a really broad set of things.
Yeah.
And I don't know. I mean, maybe it would still be able to pull something out from a less structured approach. But
Well, I think that's been something that's been bubbling on, for quite a few years now. So,
Right.
Lots of organisations have, really taken hold of this idea that data is the new oil.
Right.
Right. So all we need to do is just amass it all in a big tank. And
Right.
we can somehow sell it, and it's going to be worth, $100 a barrel or something. But it, they launch with these big aspirations for how we're going to connect all our data, but you rarely hear any success stories from that.
Right.
It hasn't really worked, and I'm wondering, how is this different?
Right. So I think for one thing, it would be about focusing it to a very specific problem area. Like I agree. Like, if you just shove a load of disparate data into one place and then somehow expect magic to happen, like somehow it's just going to fuse, it makes no sense at all. Although, yeah, again, I think people over-index on this concept of AI somehow doing something magical.
Yeah.
And in a way, like things like ChatGPT and things, because they almost feel magical, that hasn't helped things.
Yeah.
Like there's a genuine advance, without a doubt, with AI in particular. And actually, when I look at the biological space, there are particular domains where AI has done very well.
Yeah.
And they're domains with a focused problem. So say protein folding, and also a very high quality data set, which has been built up over a very long time. So I think that there is a big opportunity here, and I would like to, explore what that might mean, but also what are the characteristics of where it's worked, and how would you then apply them to this space as well? Mm-hmm. So if we take an example, say assay development, we know that we can do 1,500 runs a time when we're doing an assay dev, should we choose to. At the moment, the culture is we will do the minimum that we have to, in terms of time and experimentation and complexity of experimentation, just to get to a good enough assay.
Yeah, makes
Right?
sense.
That's kind of...
Seems reasonable.
It's reasonably reasonable. There's arguments to be made that actually we could be throwing a lot more at it to get the very best assay that we can.
Yeah.
Right, because actually then that has all sorts of knock-on implications. But that's a whole other argument, right?
Right.
And again, it's to do with data quality, but data quality that comes from that assay. Right?
Yeah, so it's kind of downstream of that project.
That kind of knock-on effect that you could have from that throughout all of discovery, which in itself is very exciting. But so that would be a minimum for me. Is okay, how do we use these 1,500 runs to get the very best assay? But then might there be a way where we have a meta experiment design? Right. So actually maybe that's not quite the right term, but it's thinking about the immediate experiment and also the ongoing experiment, if you like, that goes. So what that might look like would be, okay, well, we've got 1,500 runs, we could do a whole load of factors, right? And even if we think that some factors might not apply to this particular assay, but might apply to assays in general, then we'd deliberately include them.
Yeah.
So it'd be flat for 9 out of 10 experimental designs we do, but we're keeping it in there for consistency.
Oh, right.
Do you know what I mean?
Yeah,
So I don't know. I'm just brainstorming, right?
Yeah, well, that's the thing. There needs to be some overlap, doesn't there, between these projects for it to make sense. I mean, if these are all just isolated pockets of the biological universe, then there's no magic in AI that can somehow make some connection between these things. There does need to be that overlap, I think, and it makes me think back to my sort of process chemistry days, and if the project one day is to optimize the process of making this molecule, the next time you're making a completely different molecule, and there's people have naturally wanted to say, well, we can collect all this data, maybe it'll tell us about something about making the molecule after that.
Right.
But in reality, it's like going from, making pepperoni pizza one day to making salted caramel ice cream the next day. Right? And these are both, there's data associated with those, and they're both similar types of problems, but really you can't learn anything from the pizzas that applies to the ice cream.
Well, you certainly wouldn't want to take your temperature setting and apply it from one to the other.
Right, yeah.
You know, you'd end up with ice cream soup pretty quickly, boiling ice cream soup. Oh, that doesn't sound good. But like, I also wonder whether there's different levels of things that we can learn. Right. So, like at the most extreme level, it would be like, because we've now done 30 assays and they're all on these highly related different targets, these different proteins that are highly related to each other. So we know that they're closely related, and now we're going to do the 31st highly related one. Right?
Yeah.
Then you could maybe expect that we could make a prediction from all of those as to even the optimal assay conditions without even doing an experiment. Then we just do an experiment to confirm it, right? So that would be like the ultimate in the levels of predictivity, going almost to what, you know, you've said previously about chemistry, right? Like, oh, we think we know what should be happening, so we're going to do an experiment to confirm. Maybe we can get to those lofty heights. But actually, that's like a highly idealized system. But what we've also discussed before is about like the fact that a lot of biological systems, we don't even really know the nature of those landscapes that we're exploring. So how many factors is it useful to explore for a given like type of assay, like for a biochemical assay or a cell-based assay? Like, you know, what have the most productive optimization campaigns been? Like, you know, what kind of level of interactions did those optimization campaigns try to model for, and was it successful or not? And so, like, then it might be telling us things about the type of experimental design we're doing, even if it's not saying which factors to choose and what levels of factors to choose. And I still think that would be valuable by doing that kind of meta-analysis. Although maybe you wouldn't need AI to do that meta-analysis, actually. So,
I don't know, but I mean, that feels more realistic, and I think that's you know, that, like you say, that's still a really valuable thing, and I don't think people are really thinking that way. You know, I think that it's either they're going the whole hog towards, you know, it has to be like this in silico first idea that we're going to amass this data and just be able to make predictions forevermore, and we'll never have to do another experiment. But you know, it does seem like there's more practical, low-hanging fruit, maybe.
I think so, and I think like this is what's really valuable about this kind of conversation, because like I don't think most people would even really think about, oh yeah, it's valuable to know the type of design space you're in. But like we know that when you know that, then actually we can design a much more efficient experiment. And so then it means we're expending less resources every time to get to an answer, and maybe we're getting to a better answer as a result, because we're not missing out on like, oh, actually 3-factor interactions are really prevalent in this space, and maybe we wouldn't have tried to model those without that kind of information. I mean, like my rough mental model is, if you could imagine someone doing like hundreds of experiments across their career and having perfect memory, right? Do you think they would get benefit from all that experience or not? And if they would, then that suggests that there's some information in all of those experiments which is useful, right? And so then my guess starry-eyed vision of AI is that it's operating as that kind of collective memory. So now you've not got one person, you've got a whole group of assay scientists, maybe 40 in a company, and they're all doing an assay every 9 months. And so you're generating all, but there's no practical way of bringing all of that experience and data together in a way that's actually consumable. But maybe if we think carefully about how we're making those data sets, it could be consumable by AI, such that it could try and give us at least some more refined hypotheses that can then be tested, right?
And I think it's, you know, you're talking about this idealized scientist that's been working in the lab for 50 years and remembers everything perfectly. But I think, you know, we all, you know, every scientist does have some kind of tacit knowledge. You know, it's the things that they've learned but can't even really explain how they've learned it. They don't really necessarily know how they know it, but they know that, that's a sensible temperature range, or, you know, if it's this type of bug that I'm growing, you know, this kind of pH range is going to be reasonable. And I think, you know, that it does feel like AI should be capable of replacing that, you know, or systematizing that data into that kind of intuition.
And like, I think it can still be highly complementary as well, right? Because like people who've built up that expertise over many years will have built it up on data sets and experiments which are now in all likelihood lost to time, right?
Yeah.
But they have those mental models, and I think this is a really important point as well, that, you know, we know there's value there, and we hope that there'll be value in, you know, future AI models.
Yeah.
But really, like, the obvious thing is somehow if we can then have an interactivity between those two systems, right?
Yeah.
And so maybe you have that scientist that's having suggestions from the AI and like interrogating those, knowing their own mental models and their own heuristics of things.
Yeah.
And so in that way, you can get to the most effective next experiment.
Yeah.
Right. Which it feels like I just said that because that's what the podcast's called, but it wasn't actually meant that way at all. But like, you know, I think it's really, this is what we're after, right? Is you know, what we've been talking about is, you know, multidimensional methods are really powerful, and then we've spoken about how, you know, automation can make it still dramatically more powerful, and then we've explored a bit about what we could do with all those automation. But in the end, it's just about how are we going to be making the most powerful experiments each time we experiment in the realm of biology. And I think AI's got quite a big role to play in that in future. I suspect it does, because it's kind of got this capacity to reach. It's got much broader reach than any human does, I think.
Yeah, I think so. Yeah.
So I think, you know, we've been on quite an interesting journey thus far.
Yeah.
In this podcast. You know, we've been thinking about like the fundamental nature of biology. We've been thinking about and talking about like what our joint experience has been, which has convinced us, you know, beyond all doubt, that there are way more powerful ways of experimenting in biology than is the norm, unfortunately.
Yeah.
And then, you know, the edges of these new ways of working with automation and software, which are already reality, right? It's that classic quote of the future is here, it's just not evenly distributed. Like right now, like there are labs that are doing these very high dimensional experiments in high throughput to explore complex biology in a way that they haven't been able to before. And I find that hugely exciting.
Yeah.
And we've also, you know, spoken about how we just don't know that we're doing the best the best things with all of those runs because it's so new.
Yeah.
But I'm equally like super excited about the possibilities of then using these bigger data sets. I think, you know, to your point, it is worth adding some sobriety to this and thinking about, okay, in a fairly kind of hard-headed way about exactly what they could and couldn't do. But I do think there's a real potential there.
Yeah, and, you know, undoubtedly, collecting these high-quality, multidimensional data sets is not going to be a bad thing, right?
Right.
You know, I mean, maybe they're not going to give us that starry-eyed vision of you know, the in
solving
silico
all the
first. Yeah. But you know, in itself, it's a good idea to do your experiments smartly anyway.
Yeah.
And I always say, you know, I think what we're also talking about here is it's going to be a combination of, you know, what human understanding can bring, human intuition, experience, knowledge can bring alongside the new hardware capabilities and the new software capabilities.
Right.
So lots to explore in future episodes of the next experiment.
Yeah, but I think for today, that's where we'll wrap it up.
Lovely, thanks.
Cheers.